Imagine being born looking completely healthy. Then, before your first birthday, your body starts changing in ways we usually associate with old age. Growth slows down. Hair begins to disappear. The skin changes, the veins become more visible. The bones, blood vessels, and tissues start facing stresses they were never supposed to face so early in life. It sounds impossible, but it's real. Welcome to Science Curiosity. Today, we're diving into one of the rarest and most heartbreaking genetic disorders on Earth, progeria. The condition often described as a premature aging syndrome. But here's the first big twist. Progeria is not just one disease. Progeria includes two major disorders: Hutchinson-Gilford Progeria Syndrome, or HGPS syndrome, sometimes called adult progeria. They look similar on the surface because both create features that resemble accelerated aging, but genetically, they are not the same disease at all. Let's start with the form most people mean when they say progeria. Hutchinson-Gilford Progeria Syndrome is extraordinarily rare. This estimates an incidence of about 1 in 4 to 8 million births, with an overall prevalence of around 1 in 20 million people. Most children with HGPS appear normal at birth. Then, usually between 6 and 12 months of age, the first signs begin to appear: poor weight gain, altered skin texture, and slowed growth. And from there, the condition becomes more visible. Children with HGPS often develop alopecia, meaning hair loss, along with prominent scalp veins, pronounced eyes, a small jaw called micrognathia, and delayed dental development. As growth failure continues, the body takes on a striking shape: a thin chest, shortened clavicles, spinal curvature, and a characteristic facial appearance. But importantly, their cognitive development is generally normal. The disorder changes the body in profound ways, but it does not erase the child's mind. So what causes this? At the center of HGPS is a mutation in a gene called LMNA. This gene helps produce proteins called lamin A and lamin C, which are part of the structural scaffolding of the nucleus, the control center of the cell. In HGPS, the mutation causes abnormal lamin A, often called progerin, which distorts the nuclear envelope and destabilizes the cell. In simple terms, the cell's internal architecture starts failing, and when enough cells become unstable, tissues begin to age in ways they should not. This is why HGPS is so devastating. The most dangerous damage is not the wrinkles or the hair loss. It's what happens inside the cardiovascular system. According to the statistics, nearly 75% of patients with HGPS die from cardiovascular disease, and the median life expectancy is only about 14 years. The main threat is progressive coronary and cerebral atherosclerosis, the hardening and narrowing of blood vessels that we normally associate with old age but happening in childhood. Now here's the second form of progeria. There is also an adult form of progeria called Werner syndrome. Unlike HGPS, Werner syndrome usually doesn't show itself in infancy. People with Werner syndrome are generally normal at birth and develop normally through childhood. The warning signs usually appear in adolescence or early adulthood, often starting with a failure to undergo a normal growth spurt. Then come features that create a shocking mismatch between appearance and chronological age: gray hair, hair loss, wrinkling, sunken cheeks, short stature, muscle weakness, and a high-pitched voice. Werner syndrome is also rare, but less rare than HGPS. This estimates about 1 case per 1 million people, with a notably higher prevalence in Japan and Sardinia. More than 800 of roughly 1,000 reported cases worldwide have been identified in Japan. The mean age at diagnosis is 37 years. Werner syndrome is a completely different story. Instead of LMNA, it involves mutations in the WRN gene, also called RECQL2. This gene encodes a DNA helicase, a protein that helps with DNA and RNA replication and repair. If that repair system breaks down, genetic damage starts piling up. This genomic instability, along with the buildup of senescent cells that have stopped dividing properly, is believed to drive the premature aging features and the elevated cancer risk seen in Werner syndrome. And Werner syndrome carries a long list of serious complications. These patients can develop type 2 diabetes, atherosclerosis, mitral regurgitation, aortic stenosis, osteoporosis, and cataracts, often between ages 20 and 40. The cancer risk is also unusually high. Up to 10% of patients develop malignancies, and around 50% of reported malignancies are soft tissue sarcomas, which is not the usual cancer pattern seen in ordinary aging. Average lifespan is about 54 years, with death most often caused by cardiovascular disease, cerebrovascular disease, or malignancy. So both disorders look like aging, but they don't age every tissue in the same way, and they don't happen for the same reason. HGPS is driven by nuclear structural failure from abnormal lamin A. Werner syndrome is driven by defective DNA repair from WRN mutations. One begins in the first year of life. The other usually appears in the teen years or adulthood. One is incredibly rare, even among rare diseases. The other is still rare, but seen more often. That raises the question everyone asks. Can progeria be treated? Right now, the answer is difficult. There is no definitive cure. Management is mostly supportive, treating the complications as they appear, monitoring the heart and blood vessels, managing diabetes and lipids, addressing skin ulcers, preserving mobility, and providing genetic and psychological counseling. For HGPS, lonafarnib, a farnesyltransferase inhibitor, has shown modest but significant improvements in survival. That doesn't cure the disease, but it means the story is no longer completely hopeless, and the future may be even more hopeful. Emerging therapies under investigation, including mTOR inhibitors like sirolimus and everolimus, antisense oligonucleotides that reduce progerin levels, mesenchymal stem cell therapy, nicotinamide mononucleotide for mitochondrial function, and even CRISPR-based gene editing, which has shown promising results in HGPS models. For Werner syndrome, research directions involving mTOR inhibitors and CRISPR/Cas9-mediated gene repair. That may be the most powerful part of this story. Progeria is not just a rare disease. It is also a biological window into aging itself. These syndromes let scientists study what happens when the systems that stabilize DNA, nuclei, and tissues start breaking down too early. In other words, progeria doesn't just teach us about a rare disorder. It may also teach us something fundamental about why all humans age at all. So when you hear the word progeria, don't think of a single condition. Think of two rare genetic disorders exposing two different weak points in human biology. One in the architecture of the nucleus, the other in the machinery of DNA repair. Two different broken systems. One devastating outcome. And one of the clearest reminders that aging is not just about time, it's about what happens when the body can no longer protect itself at the cellular level. If this changed the way you think about genetics, aging, and the human body, subscribe to Science Curiosity. Thank you.